It is hopefully not going to snow, or if it is, we'll be out of here before the heavy snow falls. How's that? So thank you for rescheduling. For those of you that don't know me, I'm Dr. Sam Frank. I'm a neurologist. I'm at Beth Israel Deaconess Medical Center in Boston. I'm the director of the HDSA Center of Excellence there. Every clinic every time that I see patients now, it seems that we're talking about the news. And so, it was working before. There we go. Oh, before I get started, I just want to make sure, I'm going to be talking about a lot of different companies. As much as my teenagers would like to be able to go out and shop as much as they can because we're making all this money off of companies where I have no financial interest in any of the companies. I have done some consulting for them. And I'm also the site investigator for one of the trials. I'm not going to really talk about it. And many of the slides that I'm going to present actually come directly from Wave Life Sciences. So thank you to them for providing them. So this is what people are talking about. And it's made some unbelievable news, right? So here's BBC World News, here's CNN, and they're really enticing titles and great videos. And it's, even if nothing comes of it, it's always great to see Huntington's disease in the news and awareness and information that's put out. So. I'm going to get to what this is all about, but I want to take a step back for a second first and talk about genes and get into what we're really talking about in terms of gene-based therapy. So antisense oligonucleotide, or ASO therapies, is what's so exciting and what has been out there. This is not new technology. This is technology that's actually been around for a while. And there are 4 FDA-approved drugs that already use ASO technology. So the first one was in 1998, and that was for CMV retinitis. It's a viral infection that infects the retina, and it was the first that was on the market. Now, there was lack of demand because we had such good treatments for HIV, so this wasn't a problem, and so there wasn't a use for it, so it was pulled from the market. I hope that that's the case for medications for Huntington's disease someday. But that was the first one that was on the market. In 2008, there was another ASO therapy that was approved. That's for cholesterol that's way too high, that runs in families and causes early heart attacks. And then these two have made a lot of news recently as well. They were 2016, so a lot newer, and neurologic diseases. And as you can see, we're getting closer and closer to the brain and to neurologic diseases. And so that's why there's a lot of excitement around even the history building up to Huntington's disease. Okay, so before I get into the trials and what was out there in terms of the news, I want to go back to high school biology, or at least it's high school now. How's that? And talk about genes and proteins and stuff like that, because I think it's really important to understand what happens. So normally, DNA is in almost all of our cells. Red blood cells are the one exception, but just about every other cell in our body has DNA. And DNA is just the blueprint for building proteins. So we go from DNA, which is this 3 billion pieces of information that are squished up. It's like our card catalog in terms of our library. And then RNA is like the copier. So it really kind of takes DNA and makes it into small segments, and then RNA is really what the carpenters use to build proteins. Okay, so in our body, DNA stores the information, RNA makes it usable, and that translates into proteins. And we're really made up of proteins if we're talking about skin or digestive enzymes, cells. Everything really goes back to what DNA codes and winds up being a protein. Now, when there's a mutation in DNA is a permanent change in this sequence, and DNA is a long strand, a gene is one part of that DNA that codes for a particular protein. And so in Huntington's disease, for example, you can see here that the protein doesn't look quite right because there's too many parts that are in there, and even though there's a couple of waves here that have to curl around in order to be in about the same space. So this is what happens normally, and it happens in terms of the same sequence with an abnormal protein, but the end product may not be what is a normal functioning healthy protein. Okay, so I hope that that genetics overview of 50 years of research in about one slide is clear. I'm happy to answer questions about all this. Now, Huntington's disease, of course, this is that last slide kind of highlighted, and so if there's RNA which has come from DNA, there's a specific repeat that's here that translates into a part of that protein. Okay, and that's the same thing in terms of Huntington's disease. If there are too many repeats here, that section turns into a part of the protein that that is too big and doesn't fit and doesn't work right. So there's a problem with that expanded CAG repeat. That's so we talk about Huntington's disease, right? Too many CAG repeats. Now, taking one other step, oligonucleotides as therapeutics. And so oligonucleotides, oligo means few, and nucleotides is what we're talking about in terms of DNA and RNA. So an oligonucleotide is a short stretch of DNA or RNA that's been modified, that gets into cells and is custom built to bind to a specific sequence found in RNA. Okay, so if this is the DNA with the mutated part, here's our therapy, the oligonucleotide, that goes in and binds, in this case, to the RNA and stops the abnormal protein from being made. Okay, so that's kind of the idea. And there are lots of names that it goes by, but antisense oligonucleotide is really the full name because it's antisense, so it kind of complements the RNA strand in this case, and blocks it. You're also going to, yeah, please stop me for questions anytime.
Don't know biology much, so if you go